Okay. Round 2 Round 2. Don't worry, the final paper is easy compared to the next one. So then you summarize chapter number 7, which is on lipid. Now we look into chapter number 8, which is membrane proteins. Because we know lipids, they will merge together to create phosphoracylglycerol, and then all these lipids will form into membrane. Okay? So inside membrane, we know that they are glycoproteins, glycolipids, then we have peripheral proteins, integral proteins. Okay? So right now is that we need to understand each of the functions of the proteins. Let's start with membrane proteins. What are the two types of membrane proteins that we have? Membrane proteins. So quite you all received that? 这是什么东西来的？要下课的时候。What is that? Evacuation drill. Okay. So coming back here, the two types of membrane proteins we have. The first one is glycoproteins. The second one is glycoproteins. Okay. So what is actually the function of glycoproteins, glycolipids? They are actually responsible for cell interaction. Okay, responsible for cells interaction. And aside from that, you also need to understand about these two types of proteins, which is integral proteins. Integral and also peripheral proteins. Okay. This is integral proteins, and then we have peripheral proteins. Okay. Please understand these few kinds of membrane proteins. And then if you understand these few kinds of membrane proteins, glycoproteins, glycolipids, peripheral proteins, okay, and also integral proteins, that is where we come to the functions. What is the 6 major functions? Okay, please remember what is the 6 major functions of membrane proteins. Okay, the first one is very straightforward. It carries out transportation. The second one, not just carry out transportation. We understand that the material or the substrate entering the proteins, therefore it will convert into perhaps a product. And this process is what we call enzymatic activity. Signal transduction is signaling, cell recognition by glycoproteins, glycolipids, intercellular joining, and also last, attachment to the cytoskeleton and extracellular matrix, ECM. Okay? So if the question again asks you to, please remember the word here, if the question asks you to elaborate, it means that you need to explain. Okay, you need to explain your answer. Okay, clear? Clear? So from these 6 kinds of function, do you notice the red words here? You can explain using these red words to provide the functions of your membrane proteins. Okay? Clear? So these are the 6 functions that you need to take note of. And inside membrane, the 6 functions, okay or not? The 6 functions, clear? An elaboration of the 6 functions of membrane proteins. Okay? But what is actually more important here is also the first function, which is transportation system. We know that membrane is used for transporting substance in and out of the membrane. What are the 3 main transport systems? What are the 3 main transport systems? The first one is what we call passive transport, right? Passive transport. Passive transport is very straightforward, from high to low concentration. But if the question asks you to elaborate what is passive transport, then you need to provide a few elements. Okay, but let me finish first the transport system. The second one is facilitate transport. Okay, and the third one, which is active transport. Correct, man? Right? Correct, right? Passive, facilitate, and active. Do you all still remember I draw these 3 transport systems? Do you still remember this? Back in the class, that you need to categorize based on concentration from high to low, or from low to high. You need to justify concentration, not just only concentration. You need to know whether they have a carrier. Do you know what is a carrier? You still remember what is a carrier? This is the carrier, the channel protein, right? This kind of proteins, integral proteins, okay, the carrier, yes or no, right? What else? Does it require energy? Does it require energy? Okay. Right? These 3 types of transportation, please take note. You need to mention concentration, whether it has a carrier or not, or whether does it requires energy or not. Okay? You need to justify based on these 3 transport systems: passive, facilitate, and active. Okay? Clear? Okay? And what type, perhaps, like for example, active, what types of substrate they are transferring? Ions, right? Charge. Type. Type of molecule. What types of molecules are they transferring? Okay. And then the last but not least is this one, endocytosis. Please understand the three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, these three types of endocytosis. Okay, so this will be your fourth question, which is on your cell membrane. Okay, cell membrane. All right. Okay, I think I've already summarized all of this for you all to take note. Okay, the last chapter, which is chapter number 9. So I think we have discussed chapter number 9, right? Earlier today, earlier at 9 o'clock, our class. What are the subunits of nucleic acid? What are the subunits of nucleic acid? It's a very simple one, subunit. Understand that it has a phosphate, sugar, and a base. Okay? What are the kinds of nucleobase? Purine, pyrimidine. Okay? Purine and pyrimidine. And then the last one that you need to take note is that the complementary base pair. This part, complementary base pair. Okay, let's say given you the DNA sequence, you need to convert into RNA and then to translate into proteins. Okay, please remember that you do not need to memorize the codon table. Codon table will be provided for you. You just need to refer based on the transcripted, right? Based on the transcripted RNA. Therefore, you need to translate into the proteins. Okay? Clear? Clear? And that will be your chapter number 9 or your question number 5. That's all. Okay? Any question? Any question from the class? Anything that you want me to repeat from this chapter? Chapter 5, 6, 7, 8, 9. Which chapter do you want me to repeat? 没有了，真的是没有了，不用录了，没有了。有哪一个 chapter 你需要我再重复，还是不了解？Any chapter? No? Chapter 9. Okay. Okay, chapter 9. Let's start with part A. Very straightforward. You need to understand what is actually this structural. The whole structural is actually called nucleotide. Okay, nucleotide consists of 3 subunits. We have sugar, we have a phosphate, we have a base. Okay, and this is actually what we call a nucleic acid. Right, the whole thing is called nucleic acid, or you can call this nucleotide. Okay, and usually when we name this nucleotide, we name it based on the nucleobase. So what kind of nucleobase that we have? We have this kind of nucleobase, purine pyrimidine. Okay, purine 2 rings. 1 ring, 2 rings. Pyrimidine, 1 ring. Okay? And of course, in purine, we have adenine, guanine. Pyrimidine, we have cytosine, thymine, and uracil. Okay? But uracil only appears in RNA, whereas thymine only appears in DNA. That's straightforward. Okay? So after you understand nucleotide, nucleobase, then you need to understand what is complementary base pair. Okay? Because we know that DNA is actually double stranded. What is actually double stranded? It means that you have 2 polynuclear acid. Okay, we have 2 polynucleic acid that actually comes together based on hydrogen bond, and also the other one, which is what we call three hydrogen bond and 3 prime 5 prime phosphor diester bond. Okay, but that depends. Okay, let's say A, T, C, G. Okay, A and T, how many hydrogen bonds? You have looked into this, right? This part. A and T has two hydrogen bond. 2 hydrogen bond. Whereas C and G has 3 hydrogen bond. Still remember? Right? Still remember this, right? So let's say that this is A, this is T, then we know that this has 2 hydrogen bond. Okay? If this is G, this is C, then we have 3 hydrogen bond. Okay? But they are all linked by 3 prime 5 prime phosphor diester bond. Clear? Clear? The nucleobase only. This is only on nucleobase pairing. Okay? Clear? And this is what it means. This is 1 nucleobase. This is the second nucleobase. Okay? This is hydrogen bond. Hydrogen bond. Okay? 1, 2 hydrogen bond. How about this 1, 2, 3? 3 hydrogen bond. And that is between guanine, cytosine, and guanine. Okay? Clear? So, because the DNA has double stranded, so we will always understand that it has complementary base pairing. Okay? So let's say that if you have G, C, A, T, A, C, G, C. 1, 2, 3, 1, 2, 3, another one, let's put A. Okay? We must always read it from 5 prime to 3 prime, this direction. Okay? So the complementary of this DNA strain will always be A is T, C, G, G, C, C, G, A, T, T, A, T, C, G, and C. Clear? So, in this case, we must read this from 5 prime to 3 prime. Okay? We should not read from 3 to 5. Okay? So let's say that if you want to read it, it will always be the same. If you do this in words, it will always be the same. Okay? So once we know that we have complementary base pairing, there's always a process which is what we call central dogma of molecular biology. It means that DNA, this is DNA, not the Mayday DNA, you know Mayday have a song DNA. So anyhow, the DNA will transcript. The process is called transcription into R and A. Okay? So let's say that how does it transcript into RNA? We need to read from 5 prime to 3 prime. So we will read from this and transcript it. How are we going to transcript again? You have G, C, A, T becomes U. Okay, then we have A, C, G, C, A. Okay, in RNA, only the T becomes U. The other remains the same. Okay? So once we transcript this DNA, the last part is we need to translate into proteins. Okay? So how do we actually translate into proteins? It's based on the codon paper. Based on the codon table again, this is GAC, UAC, and GCA. Always remember, during translation process, 3 codon gives 1 protein. Okay? 3 codon gives 1 protein, or 3 nucleobase gives 1 protein. So this GCA, GCA is alanine. So we have alanine, what else? UAC, threonine. The last one, GCA, is alanine. GCA is alanine. Clear? Right? Yeah. So usually in molecular central dogma of molecular biology, we can always read it from, we can always see like A, and then you have T, and then you have A, and then this is what we call your polypeptide. Clear? Right? This is what we call your polypeptide. And it can be very, very, very long. It can be very, very long. Okay? And this is how you need to understand about this chapter. Okay? Clear? At the back? Students at the back, clear? Can score. I don't want to see you next semester already. Please graduate. Okay? So any more questions? Anything that you want me to repeat? Which chapter? Which chapter do you want me to repeat? Okay. If there's no chapter, this Friday I think we do not need to have a class, right? Or you all still want to have a class? 你们要上课吗？这个星期五？星期四你们要上课吗？因为已经教完了。Or you all want to study yourself at home? No class? Okay? Okay? So there will not be any class this Thursday. But if you have any question, you can always let me know, okay, in the group, or you can find me at my office 2409. Alright? So if there's no more question, I wish you all the best for your finals. Alright? So I do hope to see you guys next semester. Hope all of you can pass with flying colors. Okay? Alright? All the best everyone.